Welcome back to the playlist on heme synthesis and cytochrome P450. In this video, we're going to look at the penultimate step of heme synthesis, and that's catalyzed by this enzyme, which is Protoporphyrinogen Oxidase. Okay, so this enzyme, what it's going to do is it's going to do a biotransformation of protoporphyrinogen into protoporphyrin. And I know those names kind of sound similar, but what you can see basically by looking at protoporphyrinogen versus protoporphyrin is protoporphyrin actually has extensive double bond conjugation going around the whole ring. If you actually look at protoporphyrinogen doing some structural analysis, notice that right here at this point, at this point, at this point, and at this point, those don't continue on the continuous p orbital system. Okay, so when you get to that carbon, all pi system conjugation stops, and so what you have are four completely separate aromatic rings. Okay, because it's a tetrapyrrole, so there's four pyrroles, but those pyrroles are sort of independent of each other. Their pi system, their pi conjugation does not extend to the other pyrrole rings. But see, what this enzyme does is it fully aromatizes the whole porphyrin ring system. Okay, so protoporphyrin is not just it's not just four aromatic pyrrole rings. All of the pyrrole rings are now completely conjugated to each other. So this is one continuous p orbital system. It's continuous pi electron system. And what you would find is, if I actually did this with somebody one time, I was helping them with Huckel's rule for aromaticity, and I basically showed this guy that this whole ring system satisfies Huckel's rule. It satisfies Huckel's rule where you count the number of pi electrons and you set it equal to 4n plus 2. And the rule is that n has to be a whole number, and when you do this for the protoporphyrin ring, you find out that n is a whole number. Okay, so that's something really important to see. And what we might also do is let's go ahead and actually do that just to prove it to ourselves. Okay, so let me actually label the lone pairs that are going to be part of the p orbital system. Okay, the pi conjugation. So there's a lone pair here on this nitrogen. Okay. And there's also a lone pair here on this nitrogen, okay? And so what we're going to do essentially is we're going to count the number of pi electrons. And let's agree with something. Let's basically agree that this lone pair right here, let's agree that this one right here and this one are part of the aromaticity of each ring, okay? And what you'd find is that they actually are. Now, when you do this calculation for aromaticity, remember, you're only concerned about the cyclic structures, okay? So this vinyl group right here, that's not included, and then this vinyl group is not included because they're not part of the cyclic structure. So if you count them, we'll start over here. You'd have 2, here's 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, and 24, and 26 right here. Okay, so what you do to see if this satisfies Huckel's rule is what you do is you take that number, which we calculated just to be 26, right? So you say 26, which is the total number of pi electrons, right? This is the total number of pi electrons, and you set that equal to 4n plus 2. And the rule is that this n that you're going to be solving for, this n has to be a whole number. And if it's a whole number, then the whole system that you just looked at is aromatic. So let's go ahead and solve for n. So you can subtract 2 from both sides. And so you get 4n is equal to 24. And you can divide both sides by 4. And what you should see in this completely aromatic system is that when the 4's cancel, you get n is equal to 6. And certainly 6 is a whole number. So what this tells you is that this completely conjugated system of pi electrons is aromatic. So this is the first structure that we get to in porphyrin synthesis, in which there's complete pi electron conjugation about the whole porphyrin ring system. Okay, you don't see that in protoporphyrinogen because at each of these purple dots there's a discontinuity in the pi electron system. You only get rid of that discontinuity by using this enzyme protoporphyrinogen oxidase. Okay, and in this video we're going to look at its mechanism. Before we really get into the mechanism, I want to talk once again about a deficiency of this enzyme which does occur. It's called variegate porphyria. Okay, and I got some facts on it. It's very common in South Africa, apparently. About 1 in 300 people suffer from variegate porphyria. In Finland, it's not as common, but still relative to the entire Earth, it's still rather common. 1 in 75,000 people suffer from this porphyria. And just like in the case of normal porphyrias, of course, you can get things like skin damage, and also as a result of the intermediates in heme synthesis that start to accumulate, you'll get things like nausea, vomiting, constipation, all sorts of nasty things. The net root of this kind of deficiency comes from a mutation in PPOX. And PPOX is essentially the gene that codes for protoporphyrinogen oxidase. So if this guy is mutated, then essentially what happens is protoporphyrinogen 9 and everything else before it start to accumulate. And you're not going to be making protoporphyrin 9 as much. And so your heme that you biosynthesize is going to go down significantly. Okay? So another thing I want to mention about this enzyme before we actually look at the mechanism is it's a flavoprotein. Okay, this is going to be an FAD-dependent mechanism, and we're actually going to see some really cool FAD chemistry in this video. It's also an oxidase, and we looked at the last video on coproporphyrinogen oxidase, and I mentioned in that video that oxidases were enzymes that they use molecular oxygen to do a redox reaction, but none of those atoms of molecular oxygen actually get incorporated into the final product. So compare protoporphyrinogen and protoporphyrin, and you'll see there's no difference in the number of oxygen atoms. So none of these guys are actually getting incorporated into the final product. They're instead going to get lost as hydrogen peroxide, and catalysts will catabolize that. Also, one thing I did want to mention before we actually get into the mechanism of this enzyme is basically where it's located. Okay, there actually has been some furious debate over the exact location of this enzyme, but it turns out that through a series of studies that scientists have done, they've actually kind of been able to make a pretty good guess that protoporphyrinogen oxidase is located in the inner mitochondrial membrane. If you remember, coproporphyrinogen oxidase was located in the inner membrane space between the outer membrane and the inner membrane. Okay, so what ends up happening is the protoporphyrinogen that's generated in that reaction gets passed on to this inner membrane enzyme, and then that synthesizes protoporphyrin, which basically gets dumped into the matrix, where ferrochelatase will now act on it. Okay, so just want to clear that up. Now let's look at the mechanism. Okay, so here's the structure of protoporphyrinogen. Of course, we don't have that continuous pi electron system, but the very first step in this mechanism, which I'll do in green, is going to be a proton transfer. So we have this critical Bronsted-Lowry acid base residue in the active site, and we're essentially going to deprotonate right here. Okay, and that's going to force a double bond rearrangement. Okay, it's going to put these pi electrons right here. This moves over here. Now, before I show you the last electron movement, I want to have you bear in mind this, is that there's a hydride right here. In fact, there's two. Okay, so when this pi electron rearranges to be over here, these move right here, and that's going to kick off a hydride. Okay, we haven't seen too many things like this, but this hydride is essentially going to come over here, and it's going to attack this nitrogen right here on the FAD ring. That's going to force these electrons to come right here, and you're going to have these electrons come over here, and that's going to generate an oxyanion intermediate as part of the FAD. Now, before we really get into the bulk of the actual oxidation of protoporphyrinogen, I want to take a slight detour and actually look to see what happens to the FAD. Now, of course, it's FADH minus, right? I want to take a slight detour to look at that, because this process of FAD reduction, it's going to essentially happen 3 times in this mechanism. So I'll show it to you once, and then I'm going to sort of wave my hands later because I've already shown it to you. But this is going to happen essentially 3 times. Okay, so what we do is we essentially generate this FADH minus. Of course, this is the H that we're talking about right here. So let's come down here and look at it, okay? So what's going to happen is this oxyanion is going to collapse, okay? So it's going to reform the carbonyl. This double bond is going to rearrange to be over here. Now this is going to be something strange. You probably haven't seen anything like this before. This double bond right here is going to split its electrons, okay? Because remember, this double bond is a pi bond. It's 2 electrons. And it's going to do sort of like a homolytic cleavage, where one of these electrons is going to end up as a radical right here. The other electron is going to be transferred onto molecular oxygen to make a superoxide molecule. Okay, so this is a 1 electron transfer to oxygen to make this guy, which is a superoxide. Okay, this is a superoxide. Okay, and now what's going to happen is you're going to get a coupling reaction. Okay, so number one, this lone electron that's right here on the molecular oxygen is now going to couple with this radical that's on the FADH semiquinone. Okay, and that's going to create this bond right there. And then also at the same time, the negative charge on the superoxide is going to come over here and abstract a proton from the base. And to be perfectly honest, what that base is it's the same base that originally did this original proton transfer of the protoporphyrinogen ring. Okay, so that's this proton right here, this one I'm going to circle in purple, this proton right there. If we come back here to the picture, that is this proton right here. So that's the proton that was originally abstracted by the base. And now what we have is this peroxide linkage to the FAD ring. And what's essentially going to happen now is we're going to sort of get a rearrangement that's a concerted process. Now what's going to happen is something that's basically going to occur in a concerted 6-electron movement. So this lone pair is going to come in here and form a shift base. That's going to force this bond that I've highlighted in green to break, come out here and abstract that proton, okay? And that's going to generate hydrogen peroxide, okay, as shown right here. That's, of course, going to get catabolized by catalase to provide molecular oxygen and water to get rid of that guy. And that's going to regenerate the FAD ring in the completely oxidized state. So what you might see sometimes is you'll see FAD with an OX subscript. That just means it's in the fully oxidized state. And the way you recognize the fully oxidized state is this functional group right here, kind of this conjugation of shift base chemistry. Okay. So now what we've done is we've done the very first oxidation of protoporphyrinogen, and that gets us this guy right here. Now what we're going to see is sort of some really weird chemistry, actually. We're going to see some intramolecular proton transfers and rearrangements of different kinds, okay? And it's basically going to get us to where we can do the next FAD oxidation. So what's going to happen is these pi electrons right here will come and reabstract this proton, causing a double bond to form right here. Okay, so that double bond that forms is this guy right here. And that gets us to the next structure of the tetrapyrrole. Okay, now what's going to happen is this double bond right here is going to essentially break, okay, and it's going to come out and abstract this proton right here, causing this bond to break and a double bond to form right here, okay, and that gets us to this structure. And now what's going to happen is the base that was in the active site, okay, the base is now going to abstract this proton, and that's essentially going to force another double bond rearrangement. Okay, so this double bond is going to come in right here. You'll get a rearrangement like this, and once again this double bond is going to come in here, and it's going to cause ejection of a hydride. Okay, so the hydride is going to come down here, and once again it's going to attack this nitrogen on the FAD ring, causing this double bond to come in right here, this one to come right here, and you generate an oxyanion that will eventually collapse, and it will produce hydrogen peroxide using molecular oxygen, and that will be degraded using catalase, and then you'll get back to FAD. Okay, so what is the purpose of molecular oxygen in this mechanism? Okay, the purpose of molecular oxygen is simply to reoxidize FAD back to its original oxidized state, which is this guy right here. FAD is really used to perform the oxidation of protoporphyrinogen, okay, and its metabolites. But in the process, FAD gets reduced. So to reoxidize FADH- back to FAD, that's what the molecular oxygen is for, okay? So now what we get is we get this guy right here, okay? And a similar type of chemistry is going to continue, okay? This double bond is going to come out here, and it's going to abstract a proton right here, and that bond's going to break, and it's going to form a double bond right there. And that's going to get us to this structure right here. The very next thing that's going to happen is this double bond is going to come out here and abstract this proton. It's going to cause a double bond to form right here. This rearranges like that. And you can kind of see the strategy in moving these double bonds around like this. And then that double bond comes right here. Okay. And then you get this structure right here. Well, what's going to happen now? Now what's going to happen is this double bond is going to come out right here, abstract this proton, forces a double bond to form here. And then if we come over to here, we're going to do the final proton transfer with the Bronsted-Lowry base in the active site. So what's now going to happen is this base is going to deprotonate right here. That's going to force a double bond to form right here. This rearranges to be right here. This rearranges here. And then this double bond comes right here. And of course, what we have essentially is a hydride that can leave. And you guessed it, it's going to leave and it's going to nucleophilically attack the FAD that's in the active site. And once again, it rearranges to basically put the negative charge on the oxygen, forming the alkoxide or the oxyanion. Okay? And then just like we saw before, molecular oxygen is going to reoxidize the FAD- back into FAD. Okay? And in the process, after this mechanistic step, we generate the final product, which will go on to react with ferrochelatase, and that is protoporphyrin 9. Okay? So I hope this video gave you a little bit of intuition on this enzyme. In the next video, we'll look at ferrochelatase. See you in the next video.